Aloha everybody. Today my presentation will focus on amino acid degradation and more specifically on the production of urea. Most of the nitrogen in our diet is consumed in the form of proteins. Proteins are too large to be absorbed in the original form. As you know, they're polymers of amino acids with various sizes, and that is why they need to be processed by denaturation and proteolysis, which occurs in the stomach and in the small intestines. The result of that degradation is free amino acids together with oligopeptides. As you know, oligopeptides are polypeptides with a small number of amino acids, something like 10 or a few. These oligopeptides get further degraded at the lumen still of the small intestines or at the mucosa of the small intestines, and they continue to be processed even in the intestinal cell. The resulting free amino acids now are transported into the blood, and they are delivered to the liver or any other organ that requires free amino acids. When we talk about amino acid degradation, the first thing that happens with the free amino acids is the removal of the amino group eventually in the form of ammonia. That ammonia is then processed in the body, in the cell I should say, and excreted in the form of urea. The carbon skeleton of the amino acids follow different paths of degradation, of which I'm going to talk about during my next presentation. So today I will focus your attention to the processing of ammonia or the nitrogen bodies in the cell and their excretion in the form of urea. Like I said, the first step in the degradation of amino acid is the removal of the alpha amino group. And that happens following these steps. And I'll go over them in greater details in the future slides. Here I'm just outlining the steps. First is the transamination. During this process, the amino group from the free acids are transferred to alpha-ketoglutarate to form glutamate. And the reason why I've underlined it, because this amino acid will have a major role in the processing of nitrogen bodies in the cell. The next step is oxidative deamination, where the amino group is now removed from glutamate in the form of ammonia. The ammonia is then enters the urea cycle to generate urea, which is excreted. Now let's look at the different steps in a little bit more detail. So to begin, we have proteins that are proteolyzed to amino acids. These amino acids are then deaminated. And the deamination happens with the help of amino acid specific aminotransferases. The amino acid is being removed from the amino group has been removed from the amino acids and transferred to alpha-ketoglutarate. You're familiar with that metabolite. Think about where have we talked about alpha-ketoglutarate to form glutamate. The resulting product of the deaminated amino acids are known as alpha keto acids. And again, the enzymes that catalyze these reactions are known as aminotransferases. They transfer the amino group from the amino acids to alpha-ketoglutarate to form glutamate. Now, the coenzyme of the aminotransferases is pyridoxal phosphate, which is a vitamin B6 derivative. Pyridoxal phosphates temporarily binds the amino groups to form pyridoxamine phosphate. It's shown here in the structure, and it's also shown here in the figure. The aminotransferases have a diagnostic value. You may or may not have realized that, but sometimes when you do a blood test, they report these with a special acronym, and they use different acronyms based on which nomenclature they use. But nonetheless, these amino acids, these aminotransferases, I shouldn't say amino acids, aminotransferases have a diagnostic role, because if they are found in the blood, they signify tissue damage, necrosis, usually as a result of some form of muscle damage, if there's trauma, but most commonly they're used to diagnose a heart attack or liver damage like cirrhosis or some form of inflammation of the liver. So normally these enzymes shouldn't be found in the blood, but if they are found, that means that the tissues or the cells have been damaged, broken, and now the enzymes, so the cell content has been spilled in the blood, and that's how they are detected. All right, the next step is the oxidative deamination. We call it oxidative because that's the way it happens, and it's catalyzed by glutamate dehydrogenase. Glutamate is converted again to alpha-ketoglutarate to cycle back with the transamination step, but in this particular case the amino group is taken out in the form of ammonia. In this process, a pair of electrons is being picked up and loaded on NAD to form NADH. All right, the next step is now the utilization of the ammonia into the urea cycle with a final product of urea, which is then excreted into the urine. Now, I've only circled this ammonia group because you will see why a little later, because this one ammonia only contributes to one of the nitrogens of the urea molecule. All right, so what happened in this process? We took away each amino group from each amino acid, we transferred it to alpha-ketoglutarate to form glutamate, then we utilized one enzyme to pull out the amino group from glutamate to form ammonia in an oxidative way, and then that ammonia is utilized by the urea cycle to form urea, and urea is excreted. On this slide, I just wanted to generalize the production, all the sources of ammonia in the cell. We can see all the amino acids eventually load the amino group on alpha-ketoglutarate to form glutamate. Alanine here is shown separately because it plays a role in the transport of ammonia in the blood, and you'll see that in my next slide. But it also transfers its amino group to alpha-ketoglutarate to form glutamate and pyruvate. Glutamine also, and again I'll show you on the next slide, also plays a role in the transport of ammonia through the blood. And then all of them eventually will release that ammonia in the hepatocytes, and the hepatocytes will convert it to urea or other products depending on the type of animal or cell. So like I said, alanine and glutamine play a role in the delivery of ammonia to the liver. Now, it's a good place to say that the liver is the only tissue that can process ammonia and convert it to urea. The liver, again, is the only tissue that contains all of the enzymes that can convert ammonia to urea. Ammonia is a toxin, and you need to realize that ammonia cannot simply be released in the blood. High concentrations of ammonia would have a clinical representation, of which we're going to talk towards the end of this presentation, but we cannot just simply have ammonia just in higher concentration float in the cell, in the blood, I'm sorry. So there has to be a way from the ammonia to be transported from the muscles where we could use amino acids for energy, to create amino acids for energy, and somehow that ammonia has to be transported. One way is to load it on glutamate to form glutamine, and then glutamine is transported to the blood, and then from there it goes to the liver and releases its ammonia that's converted to urea. The same thing happens with the help of pyruvate. Pyruvate is a product of glucose, but if we also degrade amino acids and releasing ammonia, that ammonia is loaded on pyruvate to form alanine. Alanine then is released in the blood, which is a much safer way of transporting ammonia. Then when it gets to the liver, through alanine aminotransferase, alanine is converted to pyruvate that gets converted back to glucose and closes the cycle. But the resulting glutamate now is used to release ammonia, and from the ammonia we generate the urea. The generated urea in the liver then is a much safer form of ammonia transport. Urea now is excreted into the blood, and then from the blood it goes in the kidneys where it's filtered in the urine and released from the body. Now, different animal and different organisms have different ways of excreting their nitrogen waste. The aquatic species release ammonia. Land animals, mammals primarily, they excrete in the form of urea, and birds is uric acid. Now, in human urine, urea is holds almost 90% of all the nitrogen-containing compounds in the urine. Creatinine, you might have heard of that. Creatinine is usually used for detection of kidney function. That is about 5% of all nitrogen bodies. Ammonia is only 3%. You see, ammonia, the concentration of ammonia in the blood and even in the urine is not very high. This is a toxin. We cannot have high concentrations of ammonia in the blood. Perhaps I wanted to, maybe I should mention this next time, but I still want to point it out to you. I want to ask you this question just to think about. Think about what are the resulting products or all products of carbohydrate degradation. Think about what are they, the overall final products of carbohydrate degradation. Now think about what are the overall products of fatty acid degradation. Now think about what is the overall product of amino acid degradation. Perhaps that's not the time to really talk about it, but I just wanted you to think about that and think about which one of those three molecules is perhaps most popular in the diet world, and which one of those molecules requires a special pathway to detoxify the organism from its final products. Just think about that, and so that hopefully that will give you some perspective in terms of diet and the use of carbohydrates, fats, and proteins. We're going to talk about this a lot more in the future, but I just wanted to make that point here when we are starting to talk about the urea cycle and think about what is the purpose of urea cycle. All right. Now, the urea cycle, I just wanted to also mention something else. I was going to mention at the beginning of the lecture, but I'm going to use graphics or figures here that you're not going to find in your textbook. The reason why I'm doing that is because I kind of like these graphics a little bit better. They're a little bit clearer comparing to the ones in your textbook. So that's why I'm using. That's one of the reasons why I'm using this. So I hope this will not distract you from learning it. They are not different. They don't have any different information. The urea cycle is the same no matter where you read it. I will show you at the end the figure from the Leninger textbook so that you can see that everything is the same, just represented a little different. So don't get distracted by the fact that you will see different representation on my lecture slides compared to the textbook. The information is still exactly the same. All right. The urea cycle begins in the mitochondrial matrix, inside the mitochondria. And it starts with the conversion of a molecule of carbon dioxide. Actually, this probably shouldn't be carbon dioxide. It should be bicarbonate ion. And a molecule of ammonia with the help of 2 molecules of ATP and an enzyme called carbamyl phosphate synthetase 1. The product is 2 molecules of ADP and carbamyl phosphate. The carbamyl phosphate synthetase 1 is an enzyme that utilizes N-acetylglutamate as an activator or cofactor. There is another form of the carbamyl phosphate synthetase enzyme, and that's the carbamyl phosphate synthetase 2. We're going to talk about that enzyme when we talk about pyrimidine biosynthesis, which happens in the cytosol. So these two enzymes are located in very different places. This one's in the mitochondria. The synthetase 2 is in the cytosol, and that form does not require N-acetylglutamate. For this one, though, N-acetylglutamate is essential. It needs to be present to make that enzyme active. All right, so that's the first step. To understand better the role of ATP in this reaction, I wanted to show you this slide. It's not a simple hydrolysis. It's a complex process involving 3 intermediate reactions. As you can see, in the first one, we basically phosphorylate bicarbonate ion, forming, don't worry too much about the name, we just phosphorylate the bicarbonate. ADP is released. Now, what is the next step? The ammonia comes in and now attacks the carbon and separates the phosphate to form carbamate. Now, ask yourselves, why did we need to use ATP? We used ATP so that we can phosphorylate the bicarbonate ion, and we phosphorylate with the only reason to release the phosphate again. It's almost like, what in the world? Why did we go through this complex process so that we can just add it and then release it? Well, but if we've talked about this many times before, but I still wanted to highlight it. The principle of activation. You destabilize a perfectly happy molecule or stable molecule, such as the bicarbonate. You're adding the phosphate, making this high energy bond, which is unstable. It doesn't want to exist, but it's necessary to promote the following reaction, which is the addition of the ammonia, which normally will not be favorable. But by adding this phosphate, we're making this reaction possible. Alright, so what's the follow-up reaction? Carbamate will be phosphorylated to form carbamyl phosphate. Can you guess what the follow-up reaction would be? By adding this excellent leaving group that is perfectly fine on its own, but now is being added to a molecule that is fairly stable, such as the carbamate, what will be the follow-up reaction? If your thoughts go in the direction that there will be an attack at this carbon, your thoughts are correct. We're again preparing the molecule for the carbamate molecule for a follow-up addition by destabilizing it, by adding an excellent leaving group to its molecule. So that is the role of ATP, to destabilize, to add energy, to make reactions happen. All right. The follow-up reaction is the ornithine transcarbamylase catalyzed reaction, during which the carbamyl group from carbamyl phosphate will be added to ornithine. The product will be citrulline. You can see the carbamyl group is added now to ornithine to form citrulline. An interesting fact, ornithine and citrulline. Look carefully. What do you see? You see a carbon bound to an amino group, bound to a hydrogen, bound to a carboxyl group, and bound to an R group, a hydrocarbon that also has some other stuff on it. What does this remind you of? Same thing for citrulline, same structure. If you remember from 481, if we have a carbon that is bound to a hydrogen amino group and a carboxyl group and an R group, that's the generic structure of an amino acid. These are amino acids. I actually mentioned these two amino acids in 481 during my instructions when I told you that there are other amino acids that are not part of the protein building blocks, but still highly functional and essential for the body. And I told you then that about these two amino acids, ornithine and citrulline, and then the reason why we're never going to see these amino acids in a protein structure is because there are no corresponding codons that designate these two acids as building blocks for proteins. But nonetheless, these are very important for urea cycle, for the urea cycle. All right, ornithine picks up the carbamyl group from carbamyl phosphate, catalyzed by ornithine transcarbamylase to form citrulline. Citrulline now will be transported out of the mitochondria where the rest of the urea cycle will occur. Okay, the next step is the synthesis of argininosuccinate, synthesized by argininosuccinate synthase or synthetase. You can use either term, it's both correct. In this reaction, we use aspartate that is added to the molecule of citrulline with the help of ATP. So we basically have a fusion of aspartate with citrulline to form argininosuccinate. And you can see the aspartate molecule and you can see the citrulline molecule. This is the last time we're going to use a high energy bond. This is the last and the third high energy bond that we will use in this process. We use 2 ATPs here, 1 ATP here, but notice when the product is in a pyrophosphate. What do we know about pyrophosphate? Well, it may be stable, but it doesn't last very long. So this bond will be broken. That's another high energy bond that will be used in this process. So in essence, we've spent 4 high energy bonds in this reaction, in this cycle so far, and well, we're not going to spend any more. Like I said, so 4 high energy bonds will be spent in this cycle. All right, so how did this addition happen? Well, instead of phosphorylation, in this case, we adenylate aspartate. Well, I'm sorry, not aspartate, citrulline. And then the aspartate's amino group will attack now that carbon that was used for the adenylation reaction. The adenylate will be released, and then we end up with argininosuccinate. Again, what is the role of ATP? To add either a phosphate or the remaining part of the nucleotide to citrulline to destabilize it, form an excellent leaving group, and release AMP, and then have argininosuccinate formed. All right, the next reaction now is the conversion of argininosuccinate to arginine and fumarate. Argininosuccinase or argininosuccinate lyase is the enzyme catalyzing this reaction. Fumarate, you remember fumarate from the TCA cycle. When you compare fumarate from aspartate, you can see that basically fumarate is deaminated and oxidized aspartate. You can see the oxidation right here between the 2 carbons. And the last reaction is the breakdown of arginine, which is a normal amino acid, if you remember, L-arginine, but with the help of an enzyme called arginase, which is found almost exclusively in the liver. That's why this cycle is found nowhere else but in the liver. Then the arginase removes that top part of arginine to form urea and the resulting amino acid ornithine. If you actually look carefully, you will see that ornithine is very similar to lysine. The only difference is that lysine has 4 methyl groups right here, methylene groups, I think that's what they're called, CH2 groups instead of 3 that ornithine has. That's the only difference between ornithine and lysine. Now the urea is released, ornithine is transported back into the mitochondria, and that's how the cycle is completed. So that's basically the urea cycle. I wanted to use this slide just to illustrate where the nitrogens and where the carbon in the urea molecule come from. You can see that from the bicarbonate at the beginning, one of the nitrogens comes from ammonia and the other one comes from the amino group of aspartate. Now, even though I said that, there's actually a very good evidence suggesting that both nitrogens are actually donated from glutamate. If you think back to where we started, proteins to amino acids, and dense amino acids are transaminated, loading the amino groups to alpha-ketoglutarate to form glutamate. Glutamate then is deaminated to ammonia. However, glutamate can also be used in a aspartate aminotransferase reaction where the amino group is transferred to oxaloacetate, another well-known metabolite from the T-cell cycle, to form aspartate. Aminated oxaloacetate becomes aspartate, and that aspartate now can donate its amino group to citrulline to form, that aspartate is then added to citrulline and eventually leaves behind its amino group and gets released as fumarate. So in essence, both nitrogens could be donated by glutamate. You can see the important role of this amino acid in the nitrogen body processing. All right, this is the figure in the textbook. You might see why I didn't choose it. It's a little bit busy, but everything is the same. Here you can see the transamination reaction where we form glutamate and how glutamate enters the mitochondria and then is oxidatively deaminated to form ammonia. Ammonia then gets incorporated with the help of a bicarbonate into ATPs to carbamyl phosphate. That later on carbamyl group is transferred from carbamyl phosphate to ornithine to form citrulline. So citrulline goes out of the mitochondria into the cytosol and then accepts aspartate with the help of ATP again to form argininosuccinate. Argininosuccinate is then converted to fumarate and arginine, and then arginine is degraded to urea and ornithine, and your ornithine comes back into the mitochondria. So like I said, everything's the same. It's just that this slide was a little bit more, well, it's busier than the other ones that I used, and that's why I didn't use this as a representation. All right, the net reaction product of the urea cycle, or the next reaction I should say, aspartate, 1 molecule of ammonia, carbon dioxide or bicarbonate, 3 molecules of ATP. Product is urea, fumarate, 2 ADPs, 4 PIs. Remember, why is that? Because 1 pyrophosphate was released, which is broken down to 2 inorganic phosphates, 1 AMP, and 3 molecules of water. So we need basically 4 high energy bonds to pay for the urea cycle. The cycle itself is irreversible, and that is a big reason why the cycle is irreversible, those high energy bonds that are spent. Alright, you realize though that fumarate is one of the products. So what would happen to fumarate? Well, fumarate can be converted to malate. That malate can be transported into the mitochondria, where it gets engaged in the citric acid cycle, and from there we can, one of the products is oxaloacetate that can be converted to aspartate, and then that aspartate can be involved again in the urea cycle. This connection is known as the aspartate arginosuccinate shunt. It connects the citric acid cycle and the urea cycle through aspartate that becomes arginosuccinate. And that shunt from malate that connects, that is generated from fumarate by hydration of fumarate, and then coming into the citric acid cycle is known as the malate aspartate shunt. Here is your textbook representation. You can see clearly how fumarate is converted to malate, or either fumarate can come in or malate come in, and then aspartate is generated from oxaloacetate, and then it gets added to form arginosuccinate, etc., etc. I'm showing these other possibilities here in this graphic. I don't know to what extent these can actually happen, but theoretically, I feel like that they can happen, because oxaloacetate is a precursor for glycolysis. So in essence, if you generate excess fumarate and malate, you could generate also oxaloacetate that can form glucose. Alright, so the urea cycle happens in the liver. The produced urea is excreted in the blood and goes to the kidneys and gets excreted in the urine. Now, majority of the urea will be excreted through the kidneys in the urine. Some small part of it, though, goes through the capillaries in the intestines and enters the intestinal lumen. And that's where, obviously, we have bacteria. I'm talking about large intestines, because there's no bacteria in the small intestines. But the large intestines have bacteria, and they get degraded. That urea gets degraded to carbon dioxide and ammonia. Now, majority of that ammonia will be excreted through the stools, but some of it gets reabsorbed and enters the blood. That is the reason why we have small concentration of ammonia in the blood, because of what happens with urea in the intestines. Now, imagine that you have a problem in the kidneys, and not much of that urea can be excreted in through the kidneys. That means that the concentration of urea in the blood will increase dramatically, or to whatever extent the condition, forces that to happen. But that would lead to an increased amount going through the capillaries and entering the intestines. That means that more carbon dioxide and ammonia will be generated. Of course, more will be excreted, but also more will be reabsorbed back in the blood. Now, ammonia is toxic, and you don't want to have high concentrations of ammonia in the blood. The condition of high concentration of ammonia in the blood is known as hyperammonemia. Hyper means high. Ammonia, obviously, is the root of the word ammonia, and then emia suffix designates that this is something observed in the blood. So hyperammonemia means high ammonia in the blood. Now this is, like I said, not a very favorable condition. And there is no other way that urea or ammonia can be processed, or ammonia can be processed to urea. And if urea is not excreted in the urine, like I said, it would lead to increased ammonia, and basically it's a vicious circle that leads to eventually, based on the level of damage, to death. The symptoms of hyperammonemia are cerebral edema. That basically means swelling of the brain. Lethargy means the person is kind of tired and kind of goes on and off consciousness in a way, kind of sluggish. Anorexia, lack of appetite. Hyperventilation or hypoventilation, which means either increased breathing or decreased breathing based on the level of damage. You might want to increase your breathing, to emit more gases, I guess, if that's needed. Or sometimes the breathing center can be affected, and that may suppress breathing. Hypothermia means decreased temperature of the body. Tremors or shaking, sluggish of speech, slurring of speech, not a very clear speech, blurred vision, seizures, coma, and death. Now, hyperammonemia can be either acquired or hereditary. Acquired is when we have liver damage. That liver damage can be caused by cirrhosis, that is either caused by alcohol intoxication or chronic alcohol intoxication. It can also be caused by viral infections. Most common of those is hepatitis infections, or biliary obstruction. That's basically obstruction of the bile ducts that increases the pressure in the liver, and that eventually damages the liver. The hereditary forms of hyperammonemia are usually connected with mutations and deactivation of the enzymes of the urea cycle, and it happens with a rate of 1 in 30,000 births. Now, in each case of the hereditary hyperammonemia, basically we see the symptoms in the first weeks after birth. This is a quickly diagnosed condition because it is so important for the body to be able to process amino acids. But you can observe the symptomatic that I've talked about in the newborns even within the first 1 or 2 weeks. And all of them eventually, all of those that are as a result of deficiency of the urea cycle enzymes, they lead to mental retardation, which is most unfortunate, of course. Other problems, of course, the toxicity comes mostly because of the depletion of alpha-ketoglutarate, which obviously is going to affect ATP or energy production, which affects every single process in the body. Also, the dysregulated osmosis as a result of the increased glutamate concentrations and ammonia. That would lead to a brain swelling, which, of course, will onset all kinds of central nervous system symptomatic. All right. Now, when we talk about the urea cycle disorders and how do we manage them, we need to, it's noteworthy to say that the conditions of the urea cycle disorders are divided into 2 main groups: the proximal and the distal. The proximal involve the enzymes that are found in the mitochondria, and these are the carbamyl phosphate synthetase 1 and ornithine transcarbamylase. These 2 enzymes, if the conditions affect if one of these two enzymes are affected, the hyperammonemia is, the condition is the worst possible. The further down the cycle the problem is, the more favorable the outcome is, because more of the ammonia will be incorporated into other compounds that are water-soluble and potentially excretable, or there are other ways for them to be processed. But if carbamyl phosphate synthetase 1 is affected, that's perhaps the worst case scenario, because none of the ammonia can be incorporated into anything. And you basically, I'm sorry, I keep pointing at the bicarbonate, none of the ammonia, and that would lead to a very fast hyperammonemia. Okay, now we can use the different diagnostics, obviously, to try to figure out where the problem is. If you have high ammonia but low citrulline, obviously we cannot make citrulline, so the problem is somewhere here in these two enzymes. But if you have high ammonia and high citrulline, that means that the arginine succinate synthetase is affected, and that's why citrulline is increased. Now, if arginine succinate is high in addition to all those others, that means that the ASL enzyme is high, or the arginine succinate or arginine succinate lyase is high. These are interchangeable names. Arginine succinate lyase and arginine succinase means the same enzyme, just slightly different name. And if we have, of course, high arginine, that means that the arginine is affected, etc., etc. So, how do we manage these situations? Well, the principle is to manage these three areas. We remove ammonia, we reduce the production of ammonia, and we try to keep the physiological environment. That's the principle. Now, how we're going to do it, there's different approaches. We remove ammonia by performing dialysis or giving nitrogen scavenger drugs. I'll talk about them in the next slide. Or arginine. Now, how would arginine help this situation? And think about it. Arginine is one of the metabolites of the citric acid cycle. Why giving more arginine would help? Well, let me just say, giving more arginine will not help every single type of hyperammonemia. It will help some forms of hyperammonemia, but not all. And here how it works. If the arginase is not affected, if it's affected, then forget it. We're not going to use arginine. But if arginase enzyme is fully functional, giving arginine, what would that do? Usually you guys focus on the urea and you say, oh, it generates more urea, that helps the process. Why would generating more urea help the urea cycle? It doesn't make any sense. The focus is on the other product of arginine degradation to urea and ornithine. Now, if you, let's say, have a problem with the ASL or the ASS enzymes, and you have an increased concentration of citrulline, etc., and everything is backed up, and you still have high ammonia, but you have functional enzymes, but there's no ornithine because all the ornithine is converted into citrulline. Giving arginine, breaking it down to urea and ornithine will allow free ornithine to enter the cycle and hopefully more ammonia be loaded to it to form citrulline. Now, if the product is proximal, one of the proximal two enzymes, or proximal means kind of the upstream enzymes is affected, obviously giving more ornithine is not going to help. These two enzymes are not functional. Giving more ornithine is not going to do anything. That's why these situations are so tough to handle. But if these two enzymes are functional, and some of these, and these two enzymes now are not, arginine succinate synthetase and arginine succinase are affected, then giving ornithine will help. Giving arginine that will become ornithine will help. Urea is irrelevant in this schematic. It just gets excreted. All right, so that's about removing of ammonia. How do we reduce ammonia production? Well, we have to restrict protein intake. I'm saying restrict. Why not stop? Why wouldn't I say let's stop any protein intake? Well, because, as you know, protein is the only source of half of the amino acids we need to be able to synthesize our own amino acids. The other half can be generated. We haven't talked about this yet, but it's a good time to mention it. Half of the amino, actually you already know, 10 of the 20 amino acids are essential, and you know why they're essential, because we cannot make them. The body cannot make them, and so we have to take them with food, and the only source of these amino acids are full proteins, proteins that have all 20 amino acids. So that's why we restrict we don't stop completely. We also try to focus the energy needs of the body on carbohydrates and lipids. How do we keep physiological environment? Well, obviously proper hydration, so that we basically force the body to excrete more of the accumulated nitrogen bodies, nitrogen compounds through the urine. All right, and the last thing I wanted to talk about today are the nitrogen scavenger drugs. Nitrogen scavenger means that these drugs have the ability to bind nitrogen compounds and help them be removed from the body. That's their purpose. One of those is phenylbutyrate that can bind glutamine and help to be removed. Another one is benzoate that binds glycine and helps removing it. So these medication, these drugs, they are not a problem solver. They will not cure anybody from hyperammonemia or this condition. They're only a symptomatic treatment. They basically, we try to help the body to last as long as it can and not be hurt by high concentration of ammonia carrying compounds. I mean, if you think about it, glutamine is one of those compounds that carries ammonia from the muscles to the liver and then releases it in the liver. So binding it will help the load, the ammonia load in the liver. All right, that's all I wanted to say at this point. The remaining part of the lecture I will cover in the next presentation. The next presentation will be quite shorter compared to the others, but nonetheless, we have to talk about it. All right, I hope this made sense. As usual, if you have any questions, please email me or come and see me during my office hours. I'll be happy to help you. Thank you.